जय हिंद बच्चों. Today we will do some more topic of chapter 6, that is life processes of class 10th. So in this we'll discuss activities, right? So we'll discuss two activities. So first one is whether chlorophyll is essential for photosynthesis, right? So in this activity, first we have taken this variegated leaves. So variegated leaves means where there is somewhere they are dark green in color and at many places it's pale. So means chlorophyll is present here, right? And it is absent where the area is pale. Right? So this is called variegated leaf. And this is in potted plants. Here we have drawn one leaf, but we have taken a potted plant so that whenever we'll keep it in a sunlight, it can grow, it can do photosynthesis. It is not that we have plucked from the plant. It is on plant only, but we have not drawn it. It is on a plant, but it's a variegated leaves, right? So now what we have done, we have kept it for 3 days in dark room. So why we have done this? We have done this so that all the starch present in get utilized. So now if we'll keep it in dark for 3 days, now there will be no starch in it. Clear? After that, after keeping it for 3 days in dark room, we'll keep it in a sunlight for almost 6 hours. So what will happen? There will be some photosynthesis. So now we have to check that where this photosynthesis occur, in which area. So photosynthesis means the synthesis of starch. So we'll check whether this starch is present in the green area or all over the leaf. That we have to check. See, if it is present only in green area, it means chlorophyll is essential for photosynthesis. If it is present in all over area, it means chlorophyll is not necessarily be required for photosynthesis, right? So we need to check. So we have taken a variegated leaves, means it is dark at some places and pale at other places. See? Now we have kept it in a dark so that all starch is used up. Now we'll keep it in a sunlight for almost 6 hours. And what will happen now? The synthesis of starch. We have to check it where this starch is present. Now we have plucked one leaf and we have traced this on a sheet of paper to mark the green area, so that when we will remove the chlorophyll, we can make out the green area. Right? So we have plucked this leaf, we have traced the area on a sheet of paper. Now what will we do? See, now we will remove the pigment, that is chlorophyll, from this leaf, right? And after removing pigment, we will check the presence of starch. Clear? And then we will see where the starch is present, whether starch is present in this region only where there was chlorophyll, right? Or it is present all over area. So just to remove this chlorophyll from this leaf, what we have done, we dipped it in a boiling water. So what will happen when we put it in a boiling water? Then this membrane of the cell ruptures. And it allows the pigment to leach out of the cell. So we boil in a water for some time. Clear? And after that, we'll put this in a alcohol. And then we'll boil this in alcohol. And this alcohol, it will remove the chlorophyll. So first we'll be boiling in a water that damage the membrane of the cells and allowing the pigment to be leached out. That is, pigment is chlorophyll, leached out the cell. And then boiling in alcohol, which removes chlorophyll. So we can properly identify the starch in the leaves. Now our leaf, this leaf is there. Now we have this leaf. Clear? this leaf, it is now, this is right. So this is right. Wait, initially it was green here. Now that pigment has come out of it. Clear? Something like this was our leaf. It was green here. Now we have removed the pigment from it by boiling it in water and then boiling it in an alcohol. Clear? Now we have to check the presence of starch, where starch is present. Clear? So what we'll do, we'll put the iodine, because we know that iodine gives blue-black color with starch. So if we dip this leaf in iodine for few minutes, and then we'll take it out and rinse, then we'll see that where there was green color, that only show the blue-black region. So this is the only region which show the blue-black. Clear? This show the, it means that there is presence of starch only in this region. Clear? Or this region. And it is not in this area where there was no chlorophyll. So this activities shows that chlorophyll is essential for photosynthesis. Clear? I hope with this activity it is clear to you that chlorophyll is essential for photosynthesis. Now one more activity we'll do, and we'll see that whether carbon dioxide is essential for photosynthesis or not. Right? So for this we have taken two potted plants. That CO2 is essential for photosynthesis. So I've taken two potted plants, and we keep it in a dark for almost 3 days. So while keeping it in dark, we know that there will be no photosynthesis, and whatever starch present, that plant will use for their energy. So we'll keep it in dark so that all the starch will used up. Now hardly any starch or little starch will left. Now we'll keep each plant on a separate plate. So with one plant, we'll keep one of watch glass containing potassium hydroxide. And what is the role of this potassium hydroxide? Is to absorb carbon dioxide. So we'll keep a watch glass with containing potassium hydroxide with one plant, and we'll cover this with a bell jar and seal the bottom of the jar and glass plate with Vaseline. So we'll make this setup an airtight. So there will be no entry of air. And we have kept this potassium hydroxide in that. So that will absorb the CO2. Now in this setup, the potassium hydroxide will absorb the CO2, and there will be no entry of any air from outside. So the setup will, the inside air will have no carbon dioxide. And in second setup, we have not kept any potassium hydroxide, but we have sealed that. We have covered that plant with the bell jar and then make the setup an airtight, right? And after that, we'll keep that in the sunlight. For almost 2 hours we'll keep. Now we have to test the presence of starch, whether the same amount of starch is present in both the plants. Yeah. So for that we pluck one leaf, and that we'll do with the iodine test. Same presence of starch, and for same test we'll do with this plant leaf also, and we'll check the amount of starch. So definitely the amount of starch will be less where we have kept this setup with where we kept this potassium hydroxide. Clear? And where we have not kept the potassium hydroxide, there starch will be more. So this activity shows that there is need of carbon dioxide for photosynthesis, or CO2 is essential for photosynthesis. So I hope with these two activities you understand that chlorophyll and CO2 are essential for photosynthesis. Clear? So this is all about autotrophic nutrition for class 10th, and we'll do heterotrophic nutrition in next video. Thank you. Thank you, students.